Hello everyone, and welcome to Stem Genomics' very first webinar. I'm Annick Marcelin, Marketing Manager for Stem Genomics, and I'll be your host today. As you know, genomic abnormalities are frequent in pluripotent stem cells, and actually they may be more frequent than we think. The only way to identify them is by testing, as nothing else will help you predict them. Whether you work at research or clinical stage, you need to trust your basic material, right? The big question is how to go about it and when. Our panel of experts is going to answer this question for you today during this webinar. Since 2018, Stem Genomics has been developing solutions to help you detect abnormalities in stem cells, with the mission to increase the safety and reproducibility of your research material. We've invested in cutting-edge technologies, and for those of you who know us, you probably know our digital PCR range that has been our main technology since the company's inception. But more recently, we've also looked at next-generation sequencing. And you'll see today in this webinar why these two technologies have a lot of sense, make a lot of sense for pluripotent stem cell scientists like yourselves. Their main advantages: they enable high power detection for the CNVs and SNVs you want to detect. They're also very fast in their delivery of results, and that's particularly true for digital PCR that will give you results in just one day. At Stem Genomics, we've turned these technologies and assays into clear and straightforward reports so you can understand your results very easily. And finally, we've ensured that all the solutions were applicable to most labs. To introduce our range today, we'll have our very own Juline Vincent, our R&D manager, who's our digital PCR specialist. After some general studies in biology, Juline has developed an extensive experience expanding her biomolecular skills on the new digital PCR technologies in various biotech companies. She's joined Stem Genomics to enable us to keep on top of the latest technologies in digital PCR and ensure that we expand our range also according to your needs. And watch your space on this. That might be the subject of future webinars. Also introducing our range will be Dr. Reda Zenagi. After his PhD in molecular genetics, Dr. Reda Zenagi has been a key contributor to the Montpellier Hospital, where he specialized in next-generation sequencing and bioinformatics. He's brought to Stem Genomics this expertise and has helped us launch the StemSeq range. And he'll be presenting this to you in a few minutes. Today, you'll also hear from our guest speaker, Travis Hardcastle, Senior Product Manager at Engineer Cells from Synthego. Travis is a specialist of CRISPR and anything to do with cell line engineering. And something else that Travis likes doing is benchmarking assays. And that's what he's done with our ICS digital range, comparing it with other tests out there. But I'm not going to steal his thunder, and I'll leave him to explain this to you in a few minutes. And last but not least, Dr. Raluca Marcu, Director of Stem Cell Research at Pluristyx. Raluca received her doctorate in medical nanotechnologies from the University of Milan and several postdoctoral fellowships. And then she's been developing her skills for over 15 years in cell biology and cell metabolism. And she really knows all about iPSC lines and how to ensure they actually meet the stringent standards of therapeutic And that's what she'll be presenting for us today. She had the opportunity to test our StemSync range in its beta version as part of her quality workflow. So she'll be telling us what she thinks of it. So that's our panel for today. We hope this webinar is going to be beneficial to you all. And before we get started with the digital PCR section, I'd like to submit a quick survey to you all. You should see it appear now on your screens. Which techniques do you use to check for genomic abnormalities? You can select more than one. I'll give you a few seconds to cast your votes. I can already tell you the majority of you are voting for digital PCR, which is great. And G-banding is coming a close second, and that's not a surprise. G-banding, as we know, is the traditional method and remains a very good method to use for genomic abnormality testing. But yep, definitely digital PCR. I think we have a few, we'll be preaching to the converted, but it's now a great transition to give the floor to Gilline Vincent. Over to you, Gilline.
Hello everyone, today we'll talk about how Stem Genomics can support you in an easy CNV analysis with new digital PCR technology. But first, I have a question for you. Do you know what happens during your cell culture? It's known that human pluripotent stem cells are prone to acquire genomic alteration during that time in culture. These alterations are often recurrent, non-random, and affecting the same genomic region. Such variants are a measure of concern because they confer a selective advantage. These alterations can quickly and completely take over the culture. So it's very important to detect those alterations at an early stage and on a routine basis to ensure good quality of your cell lines and to avoid wasting time and resources on the potentially abnormal cell line. Based on these observations, Stem Genomics has performed a meta-analysis of 120 scientific publications to identify the most common abnormalities in iPSCs and build the SMART database, our proprietary database. We concluded that the most recurrent abnormalities are copy number variations, or CNV. To detect this alteration, we have developed the ICS Digital PSC test. This test is based on digital PCR technology. It contains 24 probes designed by Stem Genomics, which allows to detect 92% of recurrent abnormalities. It's ideal for in-routine control because the result is available in 1 to 3 working days. But why did we choose digital PCR? To answer this question, I will present briefly digital PCR and these advantages. At Stem Genomics, our digital PCR assays are based on the detection of several targets with TaqMan probes. First, mix of reagents and DNA sample is partitioned in nanodroplets by oil emulsion. Partitioning has the advantage of isolating the DNA target from the DNA background and to increase the PCR efficiency by bringing closer the target and the reagents to amplify it. Also, the DNA target is isolated from interfering compounds as inhibitors. The last but not the least advantage is the effective concentration. For example, for the well-known qPCR, you can analyze 2 DNA targets among 8 DNA backgrounds. But in digital PCR, you can analyze just 1 DNA target among 1 DNA background. or even you can analyze a DNA target alone in the nanodroplets, thanks to partitioning. So the limit of detection is really improved thanks to the partitioning. Moreover, the large number of partitions generated allows us to generate statistics and calculate a better confidence interval at 95% without the need to do several sample replicates. In conclusion, we save sample and time with digital PCR. Droplets are then transferred in a 96-well plate and amplified. Each nanopartition is subjected to an endpoint PCR. At the end, each nanopartition is read and sorted as negative without an amplified target, or positive with an amplified target and a specific fluorophore. At the end, digital PCR just counts each DNA isolated in nanodroplets. That's why no calibration curve is required to calculate the concentration in comparison to the qPCR. To analyze your cells, the workflow is very easy at Stem Genomics. You should provide us with cell pellets sent in dry ice, or with 40 microliters of purified DNA sent at room temperature if you are able to perform DNA extraction. Then the DNA is quantified and diluted at Stem Genomics to get an input of 5 nanogram per microliter for the digital PCR test. Stem Genomics platform team performs the digital PCR and interprets the raw data for you. A fully interpreted report is sent within 3 working days of receipt of the samples. Now, I would like to focus on the interpreted report and how to understand your results. Our interpretation method corrects CNV raw data with different coefficients. The first one is the vertical correction factor. We establish a normal data history with more than 5,000 CNV value for each probe. A correction factor is calculated for each probe and then applied to slightly correct the CNV value according to the technical history of the probe. The second factor is the horizontal factor. This factor is established according to the fluctuation observed among the 24 probes on the analyzed sample. After these two corrections, a p-value is calculated for each probe and the CNV is interpreted. In the first part of the report, there is a color tab very simple to understand. In green, the position is normal. That is to say, the CNV value is between 1.8 or 2.2, or the CNV value is outside this range, but the p-value is not significant. In purple, there is gain, and in orange, loss. Our interpretation method considers the CNV value greater than 2.2 for a gain, or lower than 1.8 for a loss, and the significant p-value. There are also trends to gain or trend to loss, which are suspected abnormalities. If the suspected CNV is an emerging recurrent variant, it will progressively take over the culture. That's why we recommend to closely monitor these samples and to testing them again a few weeks or passages later. In the second part of the report, there is a graphical view with CNV value for each position. In this example, we can see that the sample has a well-known 20Q gain abnormality and also a loss on the 18Q. There is also a trend to gain on the 1Q position, which means that few passages later or few weeks later, a gain can occur in this position. But do you know what CNV really means? In fact, the CNV value reflects the mosaicism of your cell culture. If we take the example of 20 normal cells with 2 copies, if 2 cells acquired 1 more copy, that is to say, a third copy during the cell culture, the CMV value is 2.1 and the mosaicism is 10%. If 4 cells have 3 additional copies, the CMV value is 2.2 and the mosaicism is 20%. This is the limit of detection of our ICS digital PSC24 probe test. When all the cells acquired 3 copies, the CNV value is 3.0. Then it's the same things with loss or with 4 copies and 5, and I hope it won't come to that and that you'll test your cell regularly to avoid this. With the fast turnaround time of the ddPCR, you can easily implement the ICS digital test in your cell culture process. Genetic abnormalities occur after stressful steps as reprogramming or gene editing, but also during the time in culture, where an abnormality can occur in 5 passages later only. For bank characterization, we recommend the Duo ICS Carrier test or the StemSeq panel. The Duo ICS Carrier test combines the ICS digital PSC24 probe test and a classic G-banding test to get the most exhaustive results. Thank you for your attention. I'd now like to pass the floor to Travis from Synthego, who would like to share with you his experience with the ICS digital test. I'll meet you at the end of this webinar to answer your questions. Thank you.
All right, I believe everybody can see my screen now. First and foremost, I want to thank the Stem Genomics team for allowing me to come in and speak on a topic that is very important to Synthego, and obviously it's important to this, Stem Genomics, which is just maintaining the genomic integrity of cell lines, and in our case, our biorepository of cell lines that we use to start off all of our cell lines, or all of our projects with. So I'd be remiss if I didn't take a brief moment to give a bit of a background about Synthego, who we are, and how we got started. So Synthego was a company that was formed in 2013. And it was in 2016 we really had our go-to-market with our reagents, right? This is whenever we launched our first mass-produced 100 nucleotide synthetic guide RNAs, specifically made for CRISPR, and really we're trying to create reagents at scale. It was in 2018 that we developed our multi-guide technology to really create strategies for creating highly efficient, highly effective knockouts using multiple guides mixed into one single pool. And it was also in 2018 that we decided that we were creating these reagents, so we might as well start engineering cell lines for our customers, because we already, this is something that our customers already do with the reagents. When we could, generate an automated streamlined platform similar to what we had with our reagents for our engineered cells portfolio. It was in 2018 and 2019 that we launched that out to market. Speaking more of the future and where we're going now, the company is very much moving towards a clinical side of the business and making large GP, single guide RNAs that are made specifically towards clinical trials. But that's a bit of an overview for what Synthego is and where we got started. Some of the things that we talk about here that we're really proud of are, if you start to sum together all of our edits and all of our guides from both our engineered cells and our reagents, we accounted for well over 250,000 CRISPR edits at this point. When you look at our pool efficiencies using our multi-guide technology, we are able to generate well over 80% average edit efficiencies in those pools. And particularly the things that we're really excited about is that we've been cited in well over 1,000 publications at this point, and have shipped reagents to 45 different countries across the world. So really expanding our global reach with our products and our reagents. We work with a lot of the top pharma companies out there and research institutes out there, such as St. Jude's, CRISPR Therapeutics, In Citro, or pretty much any of the top pharma clients in the world. A bit of a peek behind the curtain. This is a bit of our facility, our engineer cells facility that you see here in Redwood City, California. It's the image on the right there is what I like to creatively call, or like to call our cell nursery. This is where all of the expansion it takes place from the time we do single cell printing at 384 well plates until you get the mass number of cells we ship out to our customers. And so that's just a bit of a peek behind the curtain, just to give some visual representation of some of the automation that we have in place on our facilities. So really what we're here to talk about, though, is obviously Stem Genomics and where we see the value that they've been able to provide us as a company over the years, or most recently over this past year. Really where we see a lot of this value be driven is in our iPSC cell line editing offering. For those of you who don't know, we offer both immortalized and iPSC, knockouts, knock-ins, a wide range of clones and pools. But specifically where we see Stem Genomics playing a huge role in our platform is with our iPSC edited offering. So I'm speaking to the choir with this, I'm sure. We all know the importance, as Julian talked about earlier, the importance of assessing the genomic integrity of our cell lines. Obviously, for the reasons that she said earlier, as in culture, these cells can accumulate changes and accumulate these genomic abnormalities. These alterations in the genome can lead to changes in their gene expression or their cellular functions and things like that, and as well as increase the tumorigenic nature of these cells as they start to grow out, depending on where they pick up these genomic abnormalities. We know that, as we're all probably well aware of, there are multiple approaches for assessing the genomic abnormalities that we see within these cell lines. They all have their respective benefits as well as drawbacks. G-banding is something that was originally brought around for many decades, but obviously, as everyone knows, the resolution is very, very low with that, with the 5 to 10 megabase sort of resolution, and it's very long and it takes a lot of time. Whole human genome sequencing is a great method, right? You can get resolution down to one base pair, but it's quite costly, and not a lot of researchers can afford to do that on a large quantity of samples. A method that we used quite for a while at Synthego was the array-based method, which the curiostat method. It's great. Your resolution is getting better than what you see with G-banding, but the turnaround times can be quite significant. But what we're really here to talk about is the ICS digital, the PCS probe kits and services that we've been able to outsource through Stem Genomics as of recent. And we really find the value in this. It says a 1 week turnaround time on there, but we really see that our turnaround times are significantly less than that whenever we outsource that service. And with the 24 hotspots they've been able to develop, covering well over 90% of the common abnormalities that you see in culture. This is really where we've gone to with and pivoted our approach to measuring genomic abnormalities with our biorepositories. A bit of where this would fit within the Synthego workflow. This is an outline of that. As you can see, highlighted in red there, is we really see this from the early on side of it, from the cell qualification, as well as the QC control that we provide on the back end, right? So one of the things that's important to us is we know that whatever we put in on the front end of our automated workflows, it's going to be directly influenced by what comes out on the back end. So we want to make sure that we have high quality cells that we're starting these editing projects with, so that we can verify that we're providing high quality cells on the back end of the portfolio or of the platform. So diving into a bit of what was mentioned earlier, we benchmarked a few of the different methods out there at Synthego for determining which was the best method for us to move forward with for determining the quality of our IPS cells on platform. Here's a bit of a background of some of the cells that we have as Synthego supplied cells that we've done extensive QC on, as well as we do work with other IPS cells. So if a customer wants to provide us with their specific cell line, we can do that, and we can offer, because of Stem Genomics now, relatively quick and fast turnaround times on qualifying and the genomic integrity of those cells on platform. So a bit of a case study of some of the things that we've seen here recently, and why we've switched over to use the Stem Genomics exclusively as our provider for measuring genomic integrity. This was about a year ago. We had a customer who came to us and had mentioned to us that they had picked up a 20Q abnormality using the Stem Genomics ddPCR kit, some of the editing clones that we provided. Now, at the time, we weren't using Stem Genomics, we're using another provider for our karyotyping methodology, and this was quite a shock to us because we had tested and QC these cells before we sent them to the customer. They came back as a normal cell line. A bit of background on 20Q. Again, I'm probably speaking to the choir about this, but it's a highly popular. It's very populated. What's the word I'm looking for? It's very prominent in IPS cultures, right? And more than 20% of the cultures worldwide have this 20Q abnormality. One of the biggest things about these abnormalities is it creates a selective advantage, as we all know, and allows the cell lines to actually start outgrowing the wild type cells whenever you pick up this abnormality. So after a few passages, you will actually have your whole culture that has this 20Q abnormality present in this. Being the company that we are, we wanted to go back and internally investigate this and make sure that it wasn't something that we had, started the project with this 20Q abnormality wasn't present in our own repository of cells. This came from one of our Synthego supplied cells. So we went back and took the lowest passage parental cell line that we have from our repository to test this again, but we wanted to do a multitude of tests on this. So we were using an array-based technology at the time to pick up the genomic abnormalities. And whenever we sent this low passage cell line back out to get tested, it came back as perfectly normal using the microarray based. What was quite shocking though is that whenever we actually used the kit from Stem Genomics to test the genomic integrity of the cells, we actually were able to pick up this 20Q copy number gain with this. And so for us, this really solidified the use of Stem Genomics for doing all of our testing downstream for measuring the genomic integrity of our cells. We have worked with quite a few number of cell lines on platform here at Synthego. So what we wanted to do then was just take a look at some of the other commercially available cell lines that we'd worked with previously, and look and compare some of the different methodologies that are out there, and see if there's anything that would be picked up by the dPCR that wouldn't be picked up by any other form of method. And quite honestly, there's two other cell lines there. There's one other cell line that we had as a Synthego supplied iPSC line that we had a 20Q abnormality pop up in that wasn't present previously in the other types of tests that we did. I think what's really shocking is there's a commercially available cell line down there, number 2, that's since been discontinued, but using the ddPCR method, you were able to see that it actually had a substantial number of abnormalities that weren't being able to be picked up by either microarray or GPanning. So this is quite a shock to us, and it really, I think, solidifies the value of Stem Genomics and their methodology for being able to pick up these abnormalities. I think in conclusion, I think one of the reasons we have completely switched over to Stem Genomics, they are our partner of choice for sending out all of our cell lines to have tested before we do projects with, or on the back end of QCR cell lines. And it's really because of the targeted methodology that they use. With the 24 probes, they're able to pick up well over 90% of the common abnormalities that are present in the literature. The speed, as I said, they say a week. We know we've been getting results back within as soon as 3 days, and we send our samples out to them to have to be tested, which is really a value to us whenever we were seeing 3 to 4 weeks with our other methodology previously. The high level of confidence we have, with using the technology, with the sensitivity that you see with the ddPCR based technology. And then just the convenience, whether you do this yourself or as we do, as we outsource the service to them, it can be done with a lot of the common lab equipment that is in most labs these days. So I think with that, I'm done. Other than this is a quick sort of plug for Synthego. Right now, we do have a bit of a promo on all of our iPSC editing, whether it's knockouts, knock-ins, and really leave no variant or gene or mutation unstudied. We're happy to help you out with any of your edits moving forward. So with that, I will step back.
Thank you very much, Travis. That was really interesting. Everyone, if you have questions, don't hesitate to send them to us here and then, and we'll answer them along the way. Also, we'll have a Q&A at the end. The question section is on the right-hand side of your screen, so please, do send them our way. It makes the event more interactive. Right, so that was it for digital PCR in a nutshell. Of course, we could be dedicating so much more time to it. But now it's time to look deeper into your cells using next-generation sequencing. And for this, Dr. Reda Zenagi is going to introduce to you our latest StemSeq range. He will explain how he and his team have managed complex data that they got from sequencing, and they turn it into meaningful and digestible results for you, ready to use in your labs. Before I hand over to Reda, I would like to also suggest another quick survey. You should see it appear on your screen now. So this survey, not surprisingly, is about NGS technology. Is it something you use as part of your genomic stability QC? Okay, I'll leave you a few more seconds to enter your votes. If you don't want to vote, you can just skip the survey and that will make it disappear from your screen. Otherwise, it'll stay on your screen. Right, that's quite exciting to see that a lot of you are thinking about using NGS and not using it yet. So this next section is going to be of most interest to you all, I'm sure. I'll give the floor to Reda.
Thank you, Annick, for the introduction, and hello, everyone. I would like to give you an overview of NGS application in genetic integrity testing, and particularly how to turn a deep NGS analysis into straightforward interpretation. As Julien has previously described, human pluripotent stem cells are prone to acquire genomic alterations. During culture, these alterations could be chromosomal abnormalities, like copy number variation, or molecular abnormalities, a single nucleotide variation. Many studies have shown that single nucleotide variant could have a significant impact on the development of your cells. The most reported are those related to p53 and BCOR genes. To respond to a need for more comprehensive solutions to target both CNVs and SNVs, we have developed a high-throughput sequencing solutions called StemSeq Plus. The StemSeq Plus was designed to target both single nucleotide and copy number abnormalities. It's a robust analysis of 361 genes involved in stem cell development or in cancers. It's also a backbone of probe covering the whole genome. Its high level of sensitivity allows you to detect up to 2% of mosaicism for the SNVs and 20% of mosaicism for the CNVs. And finally, we provide you with straightforward reports for an easy interpretation. For those who want to investigate single nucleotide variant only, we have the StemSeq panel with its high level of sensitivity, since it allows you to detect up to 1% of mosaicism. The question now is, we know that the NGS is a powerful tool, but how do you manage the sheer volume of data? And to answer this question, let me show you how we at Stem Genomics turn a deep analysis into straightforward interpretation. After genomic DNA sequencing, the bioinformatics analysis is divided into 3 steps. The first step is the variant identification, where the raw data from sequencing is processed to generate a list of all identified variants present in your samples. For the SNV analysis, the list of all variants, approximately 1,000 variants, will be filtered according to the following criteria. So we exclude all variants with a low quality score. Then we exclude variants with high polymorphism in the general population. And finally, we exclude variants located in undromic positions. At the end of this process, we obtain approximately 10 variants. The last step of the SNV analysis is the most challenging one. It's the variant interpretation. At this stage, we investigate each variant separately by performing classification according to the American College of Medical Genetics and Genomics, the SEMG, then checking this classification to ensure its relevance, and finally investigating the literature to see if the variant was previously described. On the basis of all these elements, we provide you with a list of the most impactful SNV variants. For the CNV analysis, we use the same approach. All identified variants are filtered, so we exclude a variant with a high polymorphism in the general population. At the end of this step, we obtain approximately 20 variants. For CNVs, the variant interpretation are based on the SEMG classification and cross-referencing with the Stem Genomics Smart database. That will allow us to provide a summary of the most impactful CNV variants. The question is, what exactly do you get at the end of this analysis? At the end of the bioinformatics analysis, we provide you with several files. First, the raw data files. It's all data from sequencing and variant calling. These files will allow you to perform your own bioinformatics analysis, for example. Then, we provide you with files of featured data with all CNVs and SNVs identified in your sample. That allows you to do your filtering process if you want. And finally, we provide you with fully interpreted reports for both CNVs and SNVs, which we at Stem Genomics call our straightforward reports. Now, let me show you an example of our report that we offer to our clients. And we start with the SNV analysis. Here we suggest a classification of your variants from 1 to 4. 1 being variant without any impact on your cells, to class 4 representing variants with strong potential biological impact on their development. In the present example, you can see that we classify the identified variant in class 4. This variant is a stop gain variant, which means with direct consequences on protein expression of p53 gene. This variant has a allele frequency around 2.31%. This percentage represents the level of mosaicism in your sample. The variant was listed in OMIM, COSMIC, and CleanVar databases. This information is crucial since it provides the scientific proof of the variant's impact. And finally, its frequency in the general population is around 0%, meaning that the variant is a rare variant. We also give you a description of the gene as well as a summary of the variants according to the data collected previously. For the CNV analysis, only the variants with the greatest impacts are described in the reports. In this example, we identify gain on chromosome 20. This gain is present in recurrent regions listed in the SMART database and classified as pathogenic by the ACMG classification. Finally, we provide you with a summary of the SNVs and CNVs analysis. It covers all the important data to understand the behavior of your cells. So now, if you are wondering how to implement these NGS solutions in your culture process, we have some recommendations for you. You can use the StemSeq range at the cell line acquisition stage, for example, that allow you to establish the mutational and chromosomal profile of your cell lines. It is also useful at the master cell bank to identify new mutations after reprogramming or to evaluate the efficiency of your gene editing process. Finally, we recommend StemSeq at the end of the culture process for a final check of the impact of the culture process and all genetic manipulations on your cells. Thank you for your attention. Now I would like to hand it over to Raluca from Pluristyx company, who will share her experience with us with her use of StemSeq solutions, and I will answer your question at the end of the webinar. Thank you.
Thank you, Reda. Thank you, the Stem Genomics team, for having us here. It is great to be here and share some of the experiences that we had with some of the Stem Genomics assays. Just a few words before we start discussing our experience with StemSeq Plus. A few words about Pluristyx. We are a products, tools, and services company. We are located in Seattle, United States, and Toronto, Canada, and we are supporting through our offerings the development of cell therapies. Our product portfolio includes induced pluripotent stem cells, both unmodified or bearing genetic modifications, including our proprietary IACT and fail-safe gene edits. We also offer high-density cryopreserved pluripotent stem cells in the ready-to-differentiate or RTD format, and a wide range of wild-type and disease stem cell lines. In terms of services, we support clients with genetic engineering, process and analytical development, differentiation, banking, both research grade and GMP grade. As Anik was mentioning at the beginning, we had the opportunity to test Stem Genomics' newest assay, StemSeq Plus. We would love to share with you what we've learned from the testing and most importantly, how we are planning to implement this assay in our product workflow. Just a few words about Pluristyx's product workflow. Many of our pluripotent stem cell products are generated in the form of banks. I was mentioning both research use grade banks and GMP banks. And all these banks require quite extensive characterization. Just an example of banking for iPSC products. For iPSC product generation, we start with a source material bank. The source material bank is typically a somatic cell type, and we reprogram the somatic cells to induce pluripotent stem cells using a non-integrative mRNA method. The first iPSC bank that we generate, it is a small-sized iPSC bank identified as the iPSC seed bank. Samples from the iPSC seed bank are further expanded to manufacture larger size master cell banks or working cell banks. In the end, if pluripotent stem cell differentiation is of interest, we can perform further iPSC expansion and generate our proprietary ready-to-differentiate cell banks. I also wanted to mention that at any point after the iPSC seed bank generation, we do have the opportunity to introduce gene edits, including our proprietary gene edits, perform clonal selection, further expansion and banking, starting again with a seed bank, gene-edited seed bank, MCB, WCB, and so on. I wanted to also spend a couple of minutes to discuss our bank characterization approach. For bank characterization, we do perform a wide range of tests, especially to ensure cell quality and cell safety. Some of our tests that we perform include out-of-freeze recovery, sterility, mycoplasma, adventitious agent testing, and identity. And additionally, for pluripotent stem cell banks, we will evaluate cell bank pluripotency and also the differentiation potential of the pluripotent stem cells. And last but not least, I wanted to emphasize here the genetic characterization of our banks. And it is at this point that we actually take advantage of some of the tests that are available from Stem Genomics, the G-band karyotyping, and most recently, the StemSeq Plus assay. Of course, the question is, why StemSeq Plus for bank characterization? We're actually planning to use StemSeq Plus as a high-resolution complement to the traditional G-banding that everybody's familiar with. Travis touched upon the subject a little bit. I just wanted to remind everybody, karyotyping is able to detect most large numerical and structural aberrations. We're typically looking around 5 to 10 megabases and a sensitivity around 15% for cell mosaicism. However, the karyotyping will not detect small copy number variants or single nucleotide variants, and this type of variants include some of the variants that are commonly associated with pluripotent stem cell culture, and also those occurring in cancer-related genes such as TP53, for instance. StemSeq Plus, on the other hand, is able to detect both CNVs and SNVs with a much better resolution. For StemSeq Plus, we are looking at around 100 kbs for CNVs and even lower for SNVs. Another advantage that I wanted to highlight here for SNVs, the targeted analysis, as Reda was mentioning. So this targeted analysis will only detect those variants that occur at predetermined low size in genes that are known to be associated with pluripotent stem cell development and cancer. Another important feature I wanted to highlight here, StemSeq Plus will emphasize and report those variants that have deleterious or potentially deleterious biological effects, and definitely those might impact the quality and safety of our banks. This type of reporting definitely streamlines tremendously the NGS data interpretation. When do we plan to implement the StemSeq Plus in our product workflow? We are looking at implementing this assay at multiple stages during the product development and bank generation. First, we are looking at testing the source material bank, and this test will allow us to establish the genomic profile of the source material. And also another advantage is screening early on for donor materials which might present problematic genetic abnormalities even before we start with the iPSC reprogramming process. Next, of course, we want to test the iPSC seed bank. Testing the iPSC seed bank will allow us to detect any genetic changes that are associated with the reprogramming process. Everybody is aware the reprogramming process is a very complex and stressful process on the cells. During reprogramming, we deal not only with phenotypic modifications on the cells, but also with quite significant cell culture conditions that might trigger genetic abnormalities. And finally, we were planning to evaluate the genetic profile at larger bank stage, MCBs, WCBs, and RTDs. And at this stages, we're actually monitoring for emergence of genetic changes that can be associated with large-scale cell expansion. I wanted also to highlight what are the things that we love most about StemSeq Plus. Definitely the clear criteria for variant classification and data interpretation, and also the very comprehensive and descriptive data reports. I also wanted to emphasize what Reda was mentioning regarding the SNV reporting. We are only looking at those variants with potential biological impact, those that are classified as class 4 and class 3. Also those variants that are present at high frequency. Also for CNVs, we are looking at the CNVs that are located with the recurrent HPSC CNV regions, and also significantly their ACNG classification that highlights the potential to cause disease. This is quite important for CNVs. Everybody's aware sequencing will detect many CNV variants. Many of the variants actually occur naturally in the population and they do not present any pathological impact or concern. So it is important to screen them out early on. And I also wanted to share with the audience a couple of examples from our recent beta testing, highlighting the data reports. A first example here, no variants of interest were detected for both copy number variants and single nucleotide variants. This was the test that we actually performed at a pluripotent stem cell seed bank stage together with G-banding. Luckily, both assays did not really reveal any genetic abnormalities. expertise, and this will allow us to confidently proceed with further cell expansion, MCB manufacturing, WCB, and so on. This was a fortunate case scenario. However, we did experience cell lines that presented genetic abnormalities, and in this example here, StemC Plus detected several variants of concern. We have a gain-type chromosomal abnormality situated within recurrent HPSC CNV regions. We are looking here at chromosome 20, chromosome 2, chromosome 9, but also a high-frequency variant, single nucleotide variant that was detected in CHD8, which is a cancer-associated gene, and this gene might present deleterious biological effects as reported by computational evidence and was classified as class 3. So overall, to conclude, we had a great experience testing the StemC Plus. We do believe the StemC Plus assay provides valuable information for the genetic characterization of pluripotent stem cell banks, and we are really looking forward to incorporate this assay within our routine pluripotent stem cell product workflow. Thank you.
Thank you so much, Raluca, for this, for what you shared with us. And we're now going to move to the questions and answers section of our webinar. We have received a few questions, and I invite you to actually vote for them. That will help us prioritize them. I'll go through them. Yeah, I think we have a winner. Question from Julia. Can this kit be used to detect a loss of Y chromosomes? No, correct. It only probes for X's. That might be a question for Juline.
Yes, it's for me. So the ICS Digital PSC24 probe test doesn't contain the Y probe. So yes, you cannot use the PSC24 probe test to detect the Y chromosome. But at Stem Genomics, we have other tests, and I think the ICS Digital Aneuploidy test is suitable for your application. This test is not available as a kit, but only as a service. That is to say, you should provide us Stem Genomics with cell pellets or extracted DNA, and we can perform this test, so the ICS Digital Aneuploidy test, to detect the Y chromosome on your cells. So yes, it's possible, but not with the PSC24 probe, only with the ICS Digital Aneuploidy test.
Thanks for this answer, Juline. Another question from Valentine. Have you assessed or are planning to assess the ICS Digital PSC kit compatibility with other dPCR instruments than BioRads? So again, I think that's for you, Juline.
Yeah. So I know there is a lot of digital PCR platform on the market, and yes, we plan to develop our tests on another dPCR instruments than BioRads. But I cannot say which platform for the moment, but some of our clients ask us to develop on other platforms than BioRads. So yes, I think we plan to do this for the next year, but stay tuned. We share with you the news when the kit or the test will be available for another platform.
Thank you for this answer. Next question from Andrew. What are the usual amounts of SNPs and CNVs, and is this analysis enough to prove genomic stability to a regulatory body? That might be a question for Reda, if you want to take it.
Yeah. Are you hearing me? Yes,
we can
okay.
hear you.
Yeah, thank you. Sorry. Yeah, good question. I think at the moment, the cell line that we have tested until now, we identify less SNVs than CNVs. It's an agreement with the literature, since we find more publications related to CNVs and their impact on the genetic integrity of cells. So I can't give you a number of the exact SNVs that we identified on the cells that we tested in our laboratory, but I can say that the number of SNVs is less than those of CNVs. And the most CNVs are those related to the most present in and the most recurrent CNVs present in stem cells, like 20q chromosomes and the other recurrent regions.
Okay. Thank you, Reda. Scrolling through. Yeah, I can see another question from Rocio. Do you know if the FDA or European agencies are moving towards requesting this test for entering clinical trials with HPSC-derived products? Currently, the standard seems to still be G-banding. Who would like to take this one? Can be any of you, I'm sure.
I think I will start by giving my opinions about the question, because we know that currently the FDA has no specific recommendation for the NGS solution, as their focus remains on p53 genes. So they need to take into consideration these new technologies related to next-gen sequencing and all genes involved in with the significant impact on the development development of cells, and that may have a significant impact on the genetic integrity of cells. Unfortunately, today the FDA have only recommendations for p53 genes, I think.
Okay, thank you for that, Reda. Question from Cedric. Cedric mentions the article ASU&O, stem cell reports, and basically, which was at the start of the big ICS digital project for us. He's asking if an update of the recurring hotspots has been carried out. So that's the first part of the question. And he's also asking, what are the 24 hotspots positions? Perhaps Juline wants to answer this question.
Yeah, so the 24 hotspot positions are available in this article. You can find the recurrent regions and their coordinates. So yes, you can find the hotspot position in the article. Of course, we cannot say where our probes are located for confidentiality reasons. I think you understand this. But yes, there is the recurrent regions in this article. And for the update, yes, of course, our smart database are regularly updated with the last article on the subject.
Thanks, Juline. Question from Nicholas. Can ICS digital PSC be used on its own for genomic stability when publishing? Probably again for you, Juline.
Yeah. So yes, we have some clients which will publish their data with our ICS digital PSC test. So yes, it's possible. For example, in stem cell reports or stem cell research and therapy, you can publish the data with ICS digital test. but it really depends on the context and the reviewers. So if the reviewers ask you to do some debanding, it's possible, and that's why we propose also the dual ICS Cario test, which combines the ICS digital test and the debanding test to answer to help you to publish your result with digital and debanding if necessary.
Thank you, Juline. I'm afraid we have no more time for questions, but don't worry, we're going to answer all the other questions separately, just after this webinar. So we'll get back to you on these. Thank you so much for the participation, for staying till the end, and for sending your questions. It's made this event a lot more interactive. We would love to carry on the conversation. If you want, you can already get in touch with us. I think you're going to see a pop-up on the screen, and you can click there to book a meeting with our sales team. If you have questions for Travis or Raluca, do send them our way, and we will forward them accordingly. We've also prepared some resources for you. In the app section, in the handouts, you have a few PDFs you can download. So you can do that, and I'll leave the platform on a few more minutes for you to do this till the end. And you will also be able to watch the replay or send it to your teams for another month. So thank you very much. I think that's all for me today, and have a nice rest of the day, everyone.